Gareth, would you like to give us a bit of an overview about Galaxy?
Yes, thanks, Kelly, and thanks, Jeff, for setting the scene. Hello, everyone. I always enjoyed the chance to talk about Galaxy. Jeff's done a good overview of slides, so I'm really going to skip through mine and go for the always on the edge of your seat live demo option today, so you can see Galaxy working. So I'm not going to belabor the screenshot. You'll see the software in a minute. What I really wanted to highlight, Jeff's already said, and I'll stop saying that part. So Galaxy Australia has roughly 1,500 tools. It has access to more, but those tools really take their power when you combine them into workflows, and workflows continue to evolve. So this is actually quite an old workflow of about 30, 35 steps, taking multiple data types, multiple inputs, QC, mapping against the database, building a reference out of that material, and then mapping the user data onto that reference, and ultimately visualizing that. And the nice thing about it is essentially you press the play button once. Galaxy handles the scheduling of all those jobs, handles the resourcing of all that, and then you can use this workflow for one or many samples into the future. You can version control it, you can send it to your colleagues, you can attach information all around it to make it usable to yourself and others. What is behind that website, which is hosted here at ARNet in Western Sydney, are a number of computational worker nodes here, so machines to do your analysis, but also networks up in Brisbane at QCIF, Canberra at NCI, Melbourne, mostly at Melbourne Uni, and Perth at Pawsey, where we can send your jobs depending on a number of criteria. And today's talk, it's best to just think about the size of your data set. So you want to bring one genome, you want to bring 100 genomes, that's absolutely fine. We'll just send it to a destination that's configured to take that work. And at each of these destinations, we either have access to graphical processing units for particular high-end jobs, or we have high memory or high CPU machines, depending on what you need. And again, from your point of view, that is configured by us. You're always welcome to get in touch to find out about it, but there's no coding magic required by a user of Galaxy, be an early user or a power user, and we definitely have power users that are using Galaxy just as a turnkey engine to generate genome assemblies, genome annotations, proteomics, and everything else. What we do is link our service in Australia to the two other large Use Galaxy services, which are hosted out of America, mostly out of Texas, and Galaxy Europe, hosted out of Germany, where what we do is we share all the tools. So tool versions are similar across those services, and we share reference data sets. So a reference data set doesn't just necessarily mean a genome. It can be anything that you need to reference your data. We share those, and the point of sharing them is to basically take the computational load down to a third for each service, but mostly it's so researchers can share their data and work collaboratively internationally, knowing that, again, all that is taken care of, and it is identical. If you say to a colleague, go look at it on your particular Galaxy, you know the reference material will be the same. On Australia, I hinted at about 1,500 tools. We're just shy of 10,000 tools on the global toolshed, and this is well worth bearing in mind. So we host the most, I guess, commonly accessed and best practice tools, plus a certain core tool set. But if you find you're missing something, hopefully that tool already exists, and there's roughly 8,000 tools we don't have on our service. So again, when these slides are shared, please do go discover if you think you're missing that one piece in your puzzle that would link it all together. And beyond that, beyond the toolshed, if there is software which just isn't currently available for Galaxy and you'd like it to be available for Galaxy, we do have a tool request form on our landing page. Please tell us about it. Tell us about that string of R code you have, that GitHub you came across, that publication that cited something cool. We will do our best efforts to see if it can be transformed into Galaxy. For those of you that I guess identified at greater than 2 on Jeff's 1 to 5 scale, you might be interested to know that when you do run a job in Galaxy, the command line for that job is presented to you if you go looking. And we do find that this is actually a really nice transition step for those people wanting to go from GUI, graphical user or website-based bioinformatics, to command line bioinformatics. You can actually come in and see what Galaxy is doing. There are a couple of obvious hard-coded paths in here, but the rest of this syntax can be particularly useful to troubleshooting your own code writing. I just wanted to back up something Jeff said about training, and then I'll dive into the demo for a few minutes. So training is something that the Global Galaxy is really committed to. It's one of our core and very frequent activities. So a few years ago, Galaxy spun off a support service called Training Infrastructure as a Service, or TIAS. And TIAS provides for you a training queue on Galaxy, which your users register to, a particular access to servers, and this is a cluster of machines at NCI that they've been kind enough to, I guess, put towards this project for this purpose. And this gives you a high priority access to a dedicated queue of machines to allow you to run the most efficient training events that you can. And you actually get 2 different dashboards shown here on the right with an overview of what tools have been run, where a student's up to, and then a more granular, pardon me, dashboard of what tools are working and failing. Certainly in distributed training around Australia or any virtual training, I found this exceedingly useful to immediately catch errors, either errors on my behalf as the teacher, not describing something well enough, or errors on behalf of the students in a mistype. You can immediately get onto it and see that you've got errors in some of the students and address them straight away. Very, very powerful feature. So if you're considering training, please approach us about the TIAS service. And with that, I'm going to cut to a demo. I just wanted to emphasize that the Galaxy Australia team is quite a large team of infrastructure providers, support, tool developers and scientists. So you have access to all of that when you get in contact with us. I think Jeff showed enough of the training network, so I won't dive deeply into that at the moment. Won't even let it load. So here we go. Here is Galaxy Live. Here is what we call a history. So this is a collection of raw data at the bottom here, going up through processed data. And this is the assembly of a eukaryotic genome, in particular a bird species in Australia, all the way through genome assembly and genome assessment, so the QC or quality of the genome. Discovering tools is hopefully as easy as knowing the name of the tool and finding it on the list. If you don't know the name of the tool, pardon me, just kill that for a second, sorry, cancel. You can search by ontologies, and this activity will be growing in the next month or so with the next release of Galaxy, where whole domains like proteomics, genomics, multiomics will all have a domain listed here, and then you can quickly come to that domain and find out all the tools that are associated with a global ontology. What I did want to do is show you what I would like to think is how easy it is to set up a workflow, and this will be my last little demonstration. So I'm going to title this a very bland demo, just for my own purposes, and show you what it takes to create a workflow. Right. So workflows in Galaxy are presented as a canvas, so a blank canvas for which you can drag a tool onto the workflow and drag many tools and data into outputs. So for argument's sake, I am going to pick a particular tool that Jeff mentioned called Hi-Fi. I'm just going to go back one step. I want to get Hi-Fi. So Hi-Fi ASM, haplotype assembler for PacBio reads, so off the Revio, for example. I simply bring this tool into the middle of my screen. I can configure the tool and my analysis here. This configuration here becomes part of the workflow and is very reproducible. What I've noticed as I ask for my tools, there's actually a QC tool that double checks that the input data is clean. So I'm going to bring this in as a separate tool, bring it to the left, and simply I'm going to take the output of this tool and drag it to the input of this tool. This is a symbolic link that you're making visually on the screen. Under the hood, again, Galaxy is writing this as a data transformation, a folder structure, etc., a renaming exercise to move data from tool A to tool B. I need some inputs, so I can bring an input data set here. Doesn't really matter where it is on my screen. And then the last thing I'd like to do is an assessment of my assembled reads as a genome by a tool called Busco, Assembled Genomes. Busco I'm going to bring over here, and I'm going to try and pick one of these. And I say try and pick because I know that this isn't going to work. And you can see here Galaxy is flagged for me by the little orange and explanation that the output of Hi-Fi ASM is not compatible with Busco. That's okay. I need to find a solution. And the solution is listed here. This is a GFA file type. And here is a GFA to FASTA converter. Bring that in, convert my file to FASTA, pipe it into Busco, pardon me. And there we have, in a few minutes, a very basic workflow for ingesting data, for example, out of the data portal, checking the quality. We could run any number of quality checks, piping it into Hi-Fi, data transforming it, and getting a genome assessment at the end. We can do a lot more fancy things. This can actually be piped to a markdown report for digest directly as a nice visual report. I can save that and I can run it. When I run it, it's going to just take a second to invoke it. Galaxy finds for me the most reasonable set of data in my history. I know it's this particular data set here, and all I need to do is click run, and that workflow is off and running, scheduled for me and done. So that's my whirlwind tour through Galaxy. Obviously, I could talk a lot more about it, but I want to leave time for Ziad and the question and answer. Please post anything in the chat to make it, if I need to make anything clearer. Thank you, Kelly.
Awesome. Thanks, Gary.